So I'm going to talk about the 20 years of reverse genetics of coronaviruses. It's been a long way. We've done some work, but as I will show today, we have some new systems in place now that may help us in the future to really speed up reverse genetics to study coronaviruses. So, all right. So as you see here, this was about 20 years ago. That was the way how we showed our results at meetings. The old slides we had at that time, note the blue backgrounds. So essentially, to give a little bit of background, what has been done, or the timeline of coronavirus research, we know since the 1960s that we have human coronaviruses that cause usually not severe symptoms. They have been known as human coronavirus OC43, human coronavirus 229E, and they cause common cold. And there has been quite some activity in the 1980s. I think sort of a starting point was in 1980, the first international coronavirus symposium in Würzburg. And since then, this meeting has been repeated every 2 or 3 years. It's now called the Nidovirus Symposium, since we have other families now included in an order of nidoviruses. And as you see here, in 2000, so it took actually almost 15-20 years after the first infectious clone for an RNA virus for poliovirus was described, until the coronavirus came in 2000 with the reverse genetic systems. And of course, before that, you have to imagine there were years of really trying to clone this huge RNA gene. I think at least 5 to 10 years before people started to work on assembling coronavirus genomes. So essentially, the main hurdles were that an infectious clone of 30 kilobases was time-consuming and difficult to assemble. And even more important, we often saw that full-length clones, but also small pieces of cDNA, of coronavirus cDNA, were unstable in E.Coli. So that gave us a little bit of a hard time for a while, but eventually the coronavirus field came up with some unconventional systems. So these are shown here. This was all more or less in the same year, 2000, 2001, when first the group of Luis and Juanes showed that you can assemble and clone a full-length coronavirus DNA in a bacterial artificial chromosome. At the same time, or a little bit after that, Ralph Barrick came with a system different than the bug system, with a set of plasmids that contain essentially together comprise the full-length cDNA, and they were assembled or ligated together to have a full-length DNA. And we came later in 2001 with in the group of Stuart Siddell with our system, where we omitted essentially E. coli as the cloning host and cloned the entire cDNA into a vaccinia virus backbone. So this is how these different systems look like if you look more into detail. So the bug system was launched by DNA using a polymerase II promoter for transcription in transfected cells. The in vitro ligation system was based or is based on using T7 RNA polymerase promoter to make a full-length or infectious RNA that is then transfected. And the vaccinia virus system also used the T7 RNA polymerase promoter to generate the infectious RNA that was also then transfected into cells. So essentially, every system is quite unconventional compared to much easier systems in other smaller RNA virus families. So we thought about 2 or 3 years ago that there might be another way to go for this. And just to put you into the context,
well done.
so these are the heroes. So I first want to introduce Tao. So she's a PhD student who worked on the system I'm going to describe now. Nadine is a postdoc in our lab who works closely together with Tao. And then the input of this technique was done by Jörg Joris and Fabian from the Institute of Veterinary Bacteriology, just one floor down here. And they used yeast as a cloning system to modify and to clone bacterial genomes. And this is the solution. So we used essentially yeast as the cloning host. And this has been used quite a while now in the field of synthetic biology, where, as I said, for instance, mycoplasma genomes were cloned. But also more recently, large DNA viruses have been cloned in yeast, for example, the herpes simplex 1 virus. What we did initially is to essentially take our viral genome, the coronavirus genome, with about 30 kilobase pairs, kilobase in length, and divide that into pieces, so into fragments of 2 to 3 kb. And the nice thing is with the yeast that once you transform them with DNA that is overlapping, recombination will take place. And essentially you can transform them with a whole bunch of overlapping fragments. And the yeast will then take these fragments and assemble them correctly into a yeast artificial chromosome. And this is what we tried first. In this case with mouse hepatitis virus, and we used already cloned virus where we have introduced the GFP just to be able to show or to see, is that technique useful? Is it feasible? How much work is needed to get a clone? And here you see the design. So we had 9 fragments that were overlapping, and they were just simply generated by PCR from the cloned DNA. And we introduced in the primers at the genome termini small overlaps to the plasmid, to the TAR vector, and then put all that into the yeast. In the yeast, we expected that we get the yeast artificial chromosome, and from there we then screen for correct colonies, we prepare the yak, and then from there we can use the established method to rescue the virus, so meaning that we transcribe with T7 RNA polymerase and infectious RNA, put that into cells, and then in the supernatant we will have our recombinant virus. And we did that first with MHV GFP, and then we screened a number of colonies just by one PCR, and we realized they are all positive. So that was a very nice surprise. But that also meant that this sort of screen is just meaningless if all of them are positive. So we then went on to design overlap PCRs, or PCRs that span all the overlaps we have in the different between the different fragments. And we designed the PCR so that they have a different size, the PCR products. And then we could do a multiplex PCR and look for the correct sizes. Here there are 2 multiplex PCRs covering all the overlaps, and you see how efficient this cloning method is. So we don't really have any clone that is not correct. And this was, of course, very, very nice to see. That means screening is straightforward. But it was also sad because at this day, when we saw the gel, this was the last time that we ever used the Vaccinia virus system that we have used so many years. So a little bit of happy day, but also some sad day because we now had to move on to another system. So we then ask, can we also clone other viruses? So we used the MERS coronavirus and the human coronavirus 229E. Essentially did the same strategy, that we divided the genome into pieces. Again, we used already cloned DNA to just see, is MHV just an outlier, that it was so easy to get, or is that a method that can be applied to all coronaviruses? And as you can see, when we do the multiplex PCR, we again see that most clones are correct. So the system is really, really efficient for MERS coronavirus, but also for human coronavirus 229E. We then also went beyond coronaviruses to go into virus families that are also known to have here and there some problems in cloning, in maintaining a full-length cDNA stably in E.Coli. So we used here Zika virus, where we amplified fragments from viral RNA from cell culture grown virus. And as you can see here again, the assembly or TAR cloning was very efficient. And then we even moved one step further and took a clinical sample. So this was a oral nasal swab. We just knew that it's RSV. We didn't know which type, and we just used consensus primers to amplify those fragments, sampled them in yeast, and then from there on sequenced the clone and realized it's RSV-B. But again, this showed that also for other virus families where we know there are sometimes problems in cloning, that this system is quite easy to use. So this is a list of viruses we have cloned so far. So a couple of coronaviruses, also human coronavirus HKU1, for which not yet an infectious clone exists. And then later on, I will show the work on SARS coronavirus 2. Interestingly, and as I said already, you can directly clone or make an infectious clone from a clinical sample by RT-PCR, and this we could demonstrate for the human RSV-B. So the first part, infectious clone assembly, remains time-consuming, is resolved. Now the issue with instability in E. coli. What we did with MHV GFP, with the clones we obtained, and also with MERS coronavirus clones, we just passaged them. So we took 2 clones, passaged them in yeast, or passaged the yeast containing the YAGs, and sequenced at passage 1 and passage 17 or 15, and we never got any change of the cloned DNAs, telling us that this system is also very stable when it comes to the stability of the cloned DNA. Of course, we also rescued the virus. Here's the mouse hepatitis virus GFP and also the MERS coronavirus. So then the next question is, how can we use the system to modify the clones? To demonstrate this, we used the MERS clone we have obtained, and instead of modifying this clone, we just reassembled the clone again with all the fragments, but changed one fragment just by PCR to accommodate a GFP. We again transformed that into yeast and then obtained a viable virus expressing GFP. So in our hands, we think it's much easier to reassemble the clone when you want to modify something rather than modifying an existing clone, because there we have to come up with a strategy to modify this clone. But since the screening, the cloning, and propagation is so straightforward, it's very easy to, within a few days, to reassemble the whole thing and then modify by changing one fragment of choice to get a modified clone. So then, as you know, so this was essentially at the end of 2019. And then you know that SARS coronavirus 2 came, and here's a short timeline of events that you all will remember well. So it started off with this seafood market report in Wuhan, early December. There might be already some virus spread earlier. The WHO notified us on at the end of the year, and then it became essentially it went very fast until the first sequences were released on January 10th. But it took a while until actually the virus was available, and it was not available from China, but the first isolate that was obtained outside China was in Australia. And from there, it was distributed to other labs so that the research could start on this virus. So what we did then, essentially, in the timeline of the SARS coronavirus 2 reconstruction, so we, of course, wanted to apply our system to this new virus. And at the day when we saw the sequences, essentially, shortly after we ordered synthetic fragments, overlapping fragments, and then it took about 3 weeks until we had 12 or 14 synthetic fragments. There were 2 gaps. I will come to that in a minute. But we could fill them because we received at the same or a day later RNA from an isolate from Germany, and could fill those gaps by RT-PCR fragments. And from there on, we can.
2 minutes remaining.
Pardon?
2-minute warning.
Okay, 2-minute warning. Then I have to speed up. So then we essentially it took 1 week to get the virus. So I may skip a few slides. So this is essentially the rescue. So we got essentially we made the same procedure. We electroporated and then we looked in the supernatant for plaques. We had 3 versions with different 5' ends, and all of these viruses were recovered. And we also compared them to the isolate, and this with the triangles is the wild type virus that is nicely replicating like the isolate. We also did RNA-seq to see, first of all, to confirm the identity of the recovered virus. But also when you look at the read counts, you can nicely see the nested set of subgenomic coronavirus RNAs. So this is the spike. And then you go on with RNA 3, 4, 5, 6, and so on. This is a nice image to really demonstrate we have all the subgenomics in this sample. And we could also deduce the transcription signals. We also rescued a GFP virus. And this is useful for instance, for a virus neutralization assay, as is shown here, but also as it's now been used in a couple of labs to start with high throughput screening for inhibitors. Here's one example where we applied remdesivir at low concentrations and higher concentration, and it's a really nice readout, useful for high throughput screening. So with this, I conclude. So the coronavirus infectious clone can be readily assembled with a TAR cloning within a few days. They are stable. We can use the established rescue procedures from previous systems with YAG as DNA starting material, and we can easily modify those clones by new assembly of the fragments. And this is essentially the take-home message. So this is time to construct coronavirus infectious clone. So the unit is relative time unit. I will not say how many years this is, but this was me in 2000, and this is Tau now in 2020. So we could really bring down the time to clone those viruses down to a week. And here are the main players. There are many collaborators that I cannot list here, but I think all those who contributed, and particularly Tao, Nadine, and Fabian, that did the main work. So thank you very much.
Thank you, Volker, for this fascinating presentation. It's now open for questions. Rodney, if you unmute yourself, you can.
Hey, thanks, Volker, for a very nice talk. I have a question about genetic recombination, because in yeast, when the ends start to do recombination, actually sometimes the polymerases make mistakes. So did you sequence the entire 30 kilobases of DNA, and are there really 0 errors in all of those sequences?
Yes, so we of course sequence the entire cDNA. There are 0 errors. We once in a while see errors when we look at the primer sites, but this is likely, so what we see sometimes is a missing nucleotide, but this is more likely already in the primer. So the yeast is quite accurate in assembling those fragments. And we also have a few nucleotides overlap or overhang, let's say, that are not matching to the cDNA sequence between the fragments, because we cut them out from the plasmid since we have cloned them now. And this is also tolerated by the yeast if we have a few extra nucleotides that do not match.
You mentioned there were several gaps. Can you tell us what was going on there?
Which gaps do you mean?
You mentioned during your talk that there were several gaps when you were doing the cloning. Or did I get that wrong?
I'm not sure.
It gaps in the sequence from the virus when they first were identifying the virus.
I'm not sure what you mean with gaps. Maybe I misunderstand here.
Okay, we'll go on. Are there other questions for Volker?
Volker, he means that when you first started the cloning, you were missing some of the sequences, but you said that they were filled in from some other place. That's the gaps he's
from the RNA, right.
Okay. So this was the issue with the synthetic DNA that we ordered, and we had 2 fragments, fragment 5 and 7, that could not be delivered because there was the usual problem that these fragments were not clonable or difficult to clone in E.Coli. And what we used here to fill those gaps was the viral RNA that we have received in the meantime. And we just made a RT-PCR to fill that gap. And in the meantime, we have received DNA blocks and parts of the cloned fragments from the supplier, and have now assembled them also in yeast. So now they are also cloned in yeast and are stable.
And how do you deal with sequence variation, mutations in the virus as time goes on?
What we do essentially, you mean to introduce, let's say, a particular mutation in the clone?
Well, naturally occurring mutations as well.
So you mean from the ongoing pandemic, if
Yeah.
we see those, that is, yeah. So we are doing that actually right now, and we just take the corresponding fragment and modify that, and then assemble the variant virus.
Hi, Volker, this is Eric Green. I wanted to ask a quick question. How parallelizable is the process of assembly? So for example, I think you indicated it takes about 1 week to prepare one virus. Let's say you wanted to do 10 different mutants or 100 different mutants. How much time would that take?
So what we did for SARS coronavirus 2, I didn't had time to describe that. We were not sure about the 5' ends when we saw the initial sequence. That's why we made 3 versions of the 5' end. So that makes already 3 clones we actually did. And for each version, we also made a GFP. So we had 6 different clones and we all did them in parallel. So you can do up to 10 or something. How much you can handle in a BSL-3 at the same time in parallel.